Today is our second meet in the framework of this webinar. I am very happy to present a great researcher whose aim is to inspire, lead, and champion fundamental plant and crop science research for transformational yield improvements, in line with the key message of this webinar. Professor Dr. Wim van den Ende is specialized in the metabolism of soluble sugars, especially fructan, sucrose, and raffinose, family oligosaccharides related to plant biotic and abiotic stress responses. He's fascinated by the potential significance of the Fructan syndrome in the tree of life, especially in the emerging role of fructans in plant microbial interaction. He is also deeply involved into structure function relationship of sugar metabolizing enzymes. He launched the sweet immunity or sweet priming concepts into the international research community, explaining how alternative sugar signal may contribute to plant, animal, and human health and symbiotic interactions. He produced 180 peer-review papers that were cited 6,800 times, reaching more than 850 citations per year. He has a corresponding age index of 50. Various high-impact journals were tagged, such as Transimplant Science, PNAS, Biotechnology Advances, Plant Cell, Plant Journal, Plant Physiology, and New Physiology. In 2020, he was included in the prestigious list of top 1% of most cited researchers, section Crossfield Web of Science, reflecting his worldwide appreciation as a sugar and sugar enzyme specialist. He's involved in several national and international projects and also acts as editor module of Planta and Biomolecules. He teaches 12 courses related to plant science, including the module of sugar metabolism and sugar signaling in plants. Besides, he guided numerous bachelor, master, PhD students and several postdocs. I would like to thank him very especially for his talk today.
Okay. Good. Good morning, Argentina. I'm happy to be with you today. I thank Marianela for introducing me. I gave her my CV, but it was not really necessary to repeat or read it all in front of you. Anyway, my thoughts are today with Professor Giselle Martinez-Noel, who has difficulties to be with us because of her father passing away. So my condolence with this unfortunate event. Okay, today I will talk about sweet immunity in the tree of life. It's actually a title that I have constructed based on two things. One is sweet immunity principles in plants and especially fructan-based immunity. And the other is actually the presence of fructans in the tree of life. So I just merged these two parts together to this title, but for sure this is something that needs to be developed further into the future. Okay, I indeed launched the sweet immunity concept into the scientific community. But of course, others noticed these principles already long time ago. Back in 1942, I found this manuscript from Horsfall and Diamond, and they described the curious phenomenon about bull plants in tomato. These are actually plants that don't have fruits, only big leaves, and these were not susceptible to a certain foliage disease called Alternaria solani. And then they started to test a bit the reverse thing. So they removed most of the leaves of tomato plants and leave the fruit there. And then they saw the opposite, actually, that these plants were becoming highly susceptible to this particular disease. So what does this mean? It means that if you play around with source-sink relationships in plants, if you disturb them by manipulations, or of course the same can happen during certain stresses, then you can accumulate sugar in certain parts of the plants. Like for instance, if sugars accumulate in the leaves at a certain moment or during a certain timing, then it can result in less diseases. This needs to happen for a while, this sugar excess condition. And what happens is that these sugars as metabolites and as signals actually are going to lead to pathways creating antimicrobial compounds that can actually counteract the disease. And this is, of course, then in favor of the plant. So these principles are actually pretty old, but I just put this term sweet immunity on them, just clarifying that there are all kinds of mechanisms involving sugar signaling and metabolism that can change the immunity, not only of plants, but also of other organisms, like I will talk about in the next few slides. So I will kick off with the problem statement and some solutions based on sweet immunity that we can see. I will explain a bit the principles of priming and how they can boost innate immunity in plants. Then I go to fructans. What are they and how can we use them to prime plants? And what are they exactly? Are they functioning like MAMPs or rather as damps or both? And from there, it's a bit a difficult jump that I want to make because then I want to compare things a bit with the situation in animals and humans where fructans are long known as prebiotic compounds. But actually, we also know now that they can stimulate kind of signaling pathways directly in the gut system. These people working on animals and humans do not call it a priming. So they don't use the same wordings, but I want to point it out that actually it might go back to the same mechanistic ideas or principles that are underlying all these mechanisms. May be widely distributed, not only in animals, humans, but even in all eukaryotes. And maybe even behind eukaryotes. So maybe sweet immunity principles might also happen or occur even within the prokaryotic world as we know it. And then I would like to end with conclusion and perspectives. Okay, what is the problem? The problem is, of course, plant diseases. Many diseases are very widespread. We are used to using pesticides to fight all these diseases. We use 3 billion kilograms. It's a lot. This, of course, creates other problems like intoxication, deaths, and of course, leftovers of these pesticides are very bad for the environment, and they can threaten other organisms and leading to a loss of biodiversity. Of course, we all know that pathogens become resistant to some of these pesticides, and that it becomes increasingly expensive to keep up with synthesizing new synthetic pesticides. Okay. In parallel, we have a lot of animal and human diseases, not only infection diseases, but all other types of diseases that have usually to do with imbalances in reactive oxygen species and so on, like cancer. But here too, it's a bit, the same problem. It causes millions of deaths. Pathogen resistance is also into play, and we are dealing with expensive medical treatments. So also here, when we could prevent these diseases, then we would, like we do in plants, then we would progress much faster, and we could actually circumvent or find alternatives for these usually toxic pesticides that we use. Yeah. So can we use certain biologically relevant and safe priming compounds or elicitors, and that can actually used in a preventive way, both in plants but also in animals and humans? That is the question. Can we use things that actually positively influence our immune system, both in plants but also in animals and humans, making it stronger to counteract diseases? Of course, also when we make our plants healthier or keep them healthier, we can avoid pesticides residues, then we also don't eat these pesticides residues. And it's also known that they can weaken our immune system or directly cause all kinds of diseases like cancer and other stuff. Okay, so what is the solution? Is of course prevention. Both in animals and humans and in plants, there are possibilities, and there are possibilities to use the same kind of compounds. One of these compounds are carbohydrates, and that is what I want to talk or focus on today. So there are parallelisms, between animal and human vaccination strategies and priming plants, because they actually both stimulate or boost innate immunity. So when we vaccinate, we use the weakened virus or an RNA part and we inject it. But in the same way, we can spray plants with certain priming compounds, and this actually leads to similar effects, namely protecting against disease, and boosting the native immunity system of all these creatures. Okay, so the long-term objectives of the sweet immunity concept are, of course, partially replacing agrochemicals by natural and safe alternatives. Also increase biotic but also abiotic stress tolerance in plants. And so it's not only diseases that are counteracted, but in many cases such priming compounds also can fight abiotic stresses at the same time. Not always, but sometimes it goes hand in hand. So what are the options? We can do an exogenous priming, or we can make kind of transgenic plants that actually express these priming compounds from the inside out of the plant. Okay, the overall objective is, of course, to increase the health not only of plants, but also indirectly also the well-being of animals, of humans, eating these more healthy plants, but also directly by consuming the same kind of metabolites directly into their food. And then I refer to prebiotic compounds that can actively stimulate certain beneficial bacteria in the gut system. Okay, when we look at what happens in plants, I think most of you are a little bit familiar with with things like systemic acquired resistance and induced systemic resistance. On the left side here, a certain pathogen can be used to get responses in the plant. So pathogens related proteins that are accumulating, and salicylic acid is involved in these processes. Of course, this is nothing that we can apply in real life field situations, working with pathogens. So we prefer to work with ESR, induced systemic resistance, where at the root level, usually beneficial microorganisms like rhizobacteria and trichoderma are used to create systemic effects that also protect the rest of the plant. So not only the root, but also the stems and the leaves. And the hormones involved there are especially jasmonate and ethylene. Well, so besides using biological control organisms, using living microorganisms, usually they are used at the root level, but sometimes also they can be used at the leaf level. But of course, this is not always the best option and not always easy to do and in line with what farmers are used to. Farmers are, of course, more used to spraying the leaves. So there we want to get away from these sometimes toxic agrochemicals, and we want to move to natural chemicals that are not toxic and preferentially cheap, readily available, and have also a broad spectrum. And when you use them, that you also reach systemic effects, not only local effects. But most of all, they shouldn't inhibit, of course, plant growth. So if a farmer is going to use something that is natural and biodegradable, of course, there are many reasons to do so. But if that would suddenly arrest the growth of his crops, yeah, he will not spray it, of course. So that's also a very important aspect, that whatever is natural that you spray, that it does not inhibit plant growth, or only a very little bit in a very small time window, let's say. Okay, this is actually the principle of priming, and probably some of you know this graph very well, that priming usually is done at quite early stages. It can be seed or seedling or a little bit later, but you do something, you give a stimulus, a priming stimulus, and that does not come with a big cost. It's only a small cost in terms of biological energy or ATP that it costs after doing this stimulus. But then when there is a real stress coming, like a pathogen attack, then you can see that the primed plant is going to react either faster or in a more prominent way, or a combination as compared to the non-primed in blue here. And so, yes, at the end, you have a high benefit and only a low cost. So this is why priming is such interesting phenomenon. Of course, the fitness should also not be too much affected by this priming treatment. So you should be able to just have offspring of your plants after the treatment too, of course. So yes, this is the principle of priming. When you do it, you don't see big effects. It's just triggering. It's just putting things ready, but not on yet, or not fully on. Well, there is a lot of discussion about it, and some define it like, okay, it should have no cost. I'm not a believer of this. There will always be some cost in terms of ATP, but what is essential is that at the end, the benefits are actually much higher than the cost. This is what what counts actually. Okay, what happens during priming is not really known very well, but some people also at this level have been drawing comparisons between plant and animal systems, and that is very much enlightening. So what you see here is just at the left, in the green, it's a plant cell, and here in blue, this is an animal cell. And so you have some receptors there, here and here, and you have some signaling pathways that are always more or less some representatives present. So what is going to happen when you prime a plant or an animal system? Well, that you see here. So what we already know about this is that you usually, after a priming case, you don't use too much energy, but yes, you create some new things. So some receptors, a selection of them, and just a very small selection of some receptors go up, and some of these signaling pathway representatives, also the protein levels go up, but not their activity levels. So in the primed condition, they remain actually inactive. Yeah? And some things are also happening at the level of the DNA. So it means that there is already a limited DNA modification. So already epigenetics come into play under that primed conditions. So some things are changing, but altogether it's not costing too much energy. It brings the system in a ready-to-go status, in an alert status. Alert for the real stress, which comes then here. So what happens then, of course, is that these receptors already bind their ligands. Usually these are pieces of an intruder, like a pathogen. And you see that these signaling cascades get another activation. They become activated, usually by phosphorylation, dephosphorylation. And then, indeed, you get a primed defense gene expression. So some things are put on. And this is a very similar system in the animal or human context. Okay, now I was talking about priming compounds. I was already mentioning that just one class of biomolecules that is interesting are the carbohydrates. Why are they interesting? Because they are indeed cheap and readily available in big amounts and can be get out of microorganisms or plants. Today, I want to focus a bit on fructans. What are fructans? These are polymers that are built from sucrose, table sugar. And so certain enzymes are going to build these long chains. So one glucose at the end and many fructoses. And some endo-inulinases or lavonases can degrade it. And then you have some of these oligosaccharides without glucose in it. So this is actually the story of fructans. And yes, where are these fructans? Well, I think most of you know that gram-positive and gram-negative bacteria have these fructans. Many bacteria have them. They are longer than the fructans that are present in flowering plants. Not all flowering plants have them, a certain portion have them. So there are many types of fructans, but to make it a little bit straightforward and easy for you, I will just mention inulin type and levan type. There's a different linkage between the fructose units. So yes, these fructan molecules are typically reserve compounds in plants for sure. In bacteria, maybe two, although there they probably also serve more a role in protecting themselves from the environment or in interaction with other microorganisms or even other systems. So that is in full exploration, their function extracellularly in microbes. In plants, they are really inside the cells of the plants, inside the vacuoles. So they are really stored intracellularly in a dedicated organelle in the vacuole for this purpose. Nevertheless, more and more ideas are coming up that fructans can be more than just reserve compounds, but can be involved in signaling rules. Just like we know that certain plant sugars like glucose, sucrose are recognized signals. So maybe these more general signals in plant require a more specific signal for which lower concentrations are needed and which are more, let's say, depending on a very high sucrose level, because this is what how they are regulated. The genes of the fructosyltransferases are only put on high sucrose, and then the enzymes really need high sucrose to work and to polymerize. They don't work when the sucrose is too low. So maybe as such, fructan signals could be a kind of branching off of sucrose signaling pathways. With the only difference is that the fructan signals might be more specific and need just a lower threshold to be recognized. So these are ideas that are already out over a decade, and along the way there is more and more accumulating evidence that maybe indeed fructans can also act as signals in plants, but also might be emerging as other important signals among microbes or around the interactions between microbes and plants. Okay, so I come back to the title of this talk, The Tree of Life, because in collaboration with the Turkish team, with Professor Ebru Toksoy Oner, we were the first to demonstrate fructans in the archaea. And as you all know, archaea take a very central position in the tree of life. They are connected to the bacteria, but also we evolved from them. So not only fungi, but also all higher plants and animals evolved actually from an archean organism at some point. So yes, where are the fructans? So of course within the bacteria, and here within plants. Now this is not best example because maize does not have fructans, but of course there are a lot of fructan containing flowering plants. But we discovered them here. And when you go and look deeper into this archaea, this is such big part of the tree of life that has always been neglected. But now, of course, we learn to know them better because of we are sequencing all these archaea now. And you see it goes in many directions. They are splitting out, and here it links to the eubacteria, here to the eukarya. But we found these fructans here in this branch here in the hollow archaea. Historically, they were just called hollow bacteria, but these are for sure archaea, not real eubacteria. So here we found them, and these are typically organisms that we know very well to occur in extreme environments, like high salt environments, salt lakes, very salt seas, and so on. This is where we know them from. However, we learn to know that these archaea are really everywhere. Also, in our present day life, they are really everywhere. They are not only in our gut system, they are in the soil, they are in the plants, they are everywhere. And surprisingly, no one expected this, but surprisingly, if you are going to look deep into plant tissues, you find not only the expected bacilli, leaf-on producing bacilli and piney bacillus, but what you also find there are these hollow-tolerant archaea. In the middle of a fruit, for instance, you can find them. And in the beginning, everyone thought this should be an error. This is not possible. But more and more observations are made in this sense. So what does this probably mean? It probably means that these hollow archaea are not only limited to very salty environments, but are also able to thrive pretty well in high sugar environments, like at dedicated places in plant tissues. So for instance, here, this was a study on coffee fruits where they went to the complete interior of the fruit, where they sampled the microbial communities around the phloem unloading sites where it's full of sucrose. Sucrose flows out of the phloem, and there you have particular microbes that can use this sucrose and create fructans out of it. So these Halococcus, Haloferax, and so on, these organisms can produce fructans in the middle of a plant, probably where sucrose is unloading from the phloem. No one knows how on earth these organisms get there. That's a central question, but no one knows. But it seems that they are there, so it means that these are organisms that actually are adapted also to high sugar environments, and not only in very extreme environments, but also just within our plants. So this is why I want to highlight sweet immunity in the tree of life. I mean, those understudied representatives of the tree of life suddenly are in the middle of plants. So this is a very, very interesting finding, which requires a lot of further research in the future, in my opinion. So how do these things work, these fructans? If we refer to signaling, we all know the story very well about the MAMPs and the HAMPs. Those parts, biomolecules, metabolites coming directly from microbes or from herbivores. But they can, of course, also attack the cell walls of the plants, and then you get pectin fragments and other things. And they can then be considered as damage-associated molecular patterns. And all of these are, of course, sensed by certain receptors, usually present in the membrane of the plant cell. And this is then usually integrated into signaling cascades that stimulate plant innate immunity responses. So where do we are with our fructans there? Well, if this is a cell of a fructan accumulating plant and it is attacked by a pathogen, or it's bursting open because of freezing, or when it's eaten or damaged by a herbivore, then these fructans will come out, yeah, and they become present in between the cells. This is a place where they shouldn't be present. And then, yeah, this is of course weird for the neighboring cells, because the fructan should be really inside and not in between the cells. So it's considered as dangerous, and so there are probably or possibly certain receptors involved in sensing these fructans and giving these signals to increase the innate immunity of the neighboring cells, and maybe then through a cascade of mechanisms spread this information in a systemic way over a whole organ or even the whole plant. So this is, well, also what happens with ATP. Extracellular ATP is then recognized by DORN1 receptors. You probably heard about these stories. Okay, so what do we do if we spray an exogenous fructan on a plant? we just bring it in between the cells. It starts to diffuse between the cells. So yes, it can probably hit the same receptors and cause the same immune responses. What happens in a non-fructan accumulating species? Because indeed, not all plants do accumulate them. Of course, then there are no fructans, but it still means that exogenous fructan priming can work. And so it also signifies in this case, they really should be able to act like a MAMP. And so it means, and we have shown now in many non-fructan accumulators, that this system works if you spray rice as a non-fructan accumulator. It can work. Yeah. So it means that these receptors are probably meant to be primarily against fructans from exogenous origin. So it means against bacterial fructans that it was invented during the course of evolution. In fructan accumulating plants, probably there was a further evolution of such receptor in those that recognize bacterial fructans and those that recognize their own fructans if they leak out of the cells. Anyway, you can read more about this in this manuscript if you want. So this is only one theory, because another theory is exactly the same principles or theory that happens when we eat fructans. When we eat fructans, we create prebiotic effects, as I said, in the gut system. Maybe very similar mechanisms are into play when we spray fructans onto the leaf of a plant. So maybe these fructans can be used as a food to stimulate certain beneficial bacteria so that they get the overhand over those that cannot use these fructans. So if you create a multiplication of beneficial bacteria, these pink ones, and they make elicitors, and these elicitors are actually sensed by the plant cells, then you have a completely different mechanism to explain how or what can happen if you spray fructans onto a plant. So you can have direct mechanisms that these fructans or part of these fructans, a small fructan oligosaccharides, are directly sensed by certain receptors. Or you can have an indirect effect that through the multiplication of beneficial bacteria, you actually make certain elicitors that actually then stimulate plant immune signaling and defense pathways. Okay, we are, of course, interested not only in the fundamentals, but also bring it into practice. And I think this is one of the reasons why I was invited here, because it indeed reflects the goals of this webinar. So one of our favorite system is the pathos system consisting of lettuce and Botrytis cinerea. It's a fungus that creates a lot of problems in a wide array of crop species. So this is why we also focus on that one. So what we usually do is we prime on attached leaves of the lettuce. We wait a number of days and then we cut off the leaf and we infect and we bring it into a humid environment. And then we do a disease scoring, just measuring the lesion sizes of these developing infection. So we usually work with several disease classes, as you can see here. The more black, the bigger the spots. So what you can see here is a priming with water, oligogalacturonides, pectin fragments. This is a typical damp. And then two types of fructans, inulin and livon. And you see that indeed, especially these more bigger spots are really going down when you prime the plants with inulins or livons, the lettuce. So, well, Here you see some profiles of chicory, inulin, and leaf-on from dactylis. So that's a plant origin. You see this DP, this degree of polymerization is a little bit higher, but it's essentially another type of fructan. And you see that the leaf-on is performing still a little bit better than the inulin type. Here you see just these spots developing after a few days. And you see that there are nearly no spots in the inulin and leaf-on condition. So here is another kind of control, an osmotic control. You see a lot of infection, a big lesion area. And with these pectin fragments, you have a good effect, but it's even stronger with inulin, as you can see here. And also with different kind of leaf-ons, and especially the higher levels, higher concentration. So this is 5% leaf-on, this is 1% leaf-on, are doing well. Also, GABA is an interesting compound, which is a bit similar to BABA. BABA is actually a typical priming compound that is also used in practice, but also GABA is a good priming compound, comparable to inulin in the lettuce botrytis pathosystem. Well, we tested many species, a too long list to actually make here, but some of the published ones here is not against botrytis, but against apple scab. And there, interestingly, we not only discovered a plant protective effect of this sweet priming with fructans, but also a direct effect on the development of this fungus. So the germination and the growth of this fungus itself was also affected by the leaf-on type fructans, but not by the inulin type fructans. So this indeed tells that there is a lot of variation how different pathosystems react on different types of fructans. So yeah, it means that there are probably it's probably a game of different receptors or other mechanisms that we still do not fully understand. Okay, this is a comparison with different fractions of leaf-on. What we actually did, we took Halomonas leaf-on, a gift from our collaborator in Turkey, and we used an endoleafonase to split it into different DP fractions. And so what you can see here, that is fraction 3. It's actually a DP between 3 and 9 approximately, that you can see here. So this was fraction 1, only very high DP fructans here, this peak in the back, then an intermediate fraction, and then this lower DP fraction. And you see here that this lower DP fraction behaves very well, and even almost equally well than Aliat, which is a real fungal a fungicide that is commercially available and widely used. So yes, this is a natural thing, and it's performing quite well as compared to a commercial fungicide. So yeah, here's some pictures again of these different fractions of these leaf-on oligosaccharides and intermediate and higher DP leaf-on. So this is the fraction 1, 2, 3, and you see that these lesions go down. Here you see almost no lesions, and this is very comparable to this situation here, which is Aliat, the commercial fungicide. So of course, this was done, well, I didn't mention, but this was done on rucola. Rucola is a sister from Arabidopsis, which is of course a model plant. So we also had a look at Arabidopsis because we want to go deeper into the mechanistic pathways in the future by using or exploring Arabidopsis mutants. So this is now the first phase on priming with fructans in Arabidopsis botrytis pathosystem that was published recently in Antioxidants what you see here, we compared unprimed, water primed, and then a lot of different fructan types. And then this is BABA. This is a widely used priming compound for comparison. So what you see is that if you focus on these white things, especially, yeah, you really see that these Levan oligosaccharides between 3 and 9 are behaving better than the higher DP Levans and also better than the inulin type fructans. So yes, at a certain moment, these shorter degree of polymerization Levan oligosaccharides became our lead compound to be used in other systems as well. Usually, the reactions of plants on certain elicitors can be followed because they produce a ROS burst. And what you see here is when you compare BABA with different concentration levels of these Levan oligosaccharides, you only see a ROS burst with these OGs. These are pectin fragments. So yes, here they act actually as elicitors, but real priming compounds do not cause a kind of effect when you use them. So this shows that these Levan oligosaccharides are not causing a kind of alert situation and a real stress response yet. No, they don't, but they are priming. That you can see here when you do the priming with this loss with bacterial long DP Levans or with plant Levans. And you have it followed by FLAG22 treatment. This is a typical treatment that is causing a high stress response on plants. So when you prime it first with these fructans, then you see, as compared to the water, that you get a much higher ROS peak when you use these Levan oligosaccharides before using this FLAG22 treatment. So we learned from this that, well, we learned two things. Loss is the superior fructan, Levan oligosaccharides, and yes, we have evidence that these compounds are not acting like elicitors, already putting plants in alarm and consuming a lot of ATP. No, they are acting as priming compounds, making plants ready for more serious stress signals to come, so they can either react quicker or in a more strong way. Okay, this is a very important new paper that I have to mention, because this is about the novel fructan detour concept, which was introduced by a Chinese team. And I consider this as a special case of the sweet immunity. We all know that plants exude through their roots a lot of sugar. Sucrose is the main compound there that they exude, next to some other molecules like acids and so on. But sucrose is a very important compound. And we all know that Bacillus subtilis and other beneficial bacteria transform this sucrose into fructans, usually Levan type of fructans. And this group described now that when the Levan is degraded into this loss, these Levan oligosaccharides, they too believe these are really the signals that are responsible for surfactant production by this beneficial bacteria. And this surfactant can have different effects, not only stimulating the bacterial motility so that they can more easily colonize the root, but also surfactant can directly trigger some immune pathways in plants. So all this together, this root colonization and direct effects on immunity, can increase plant immunity and can counteract the attack of pathogens. Of course, if the whole roots are surrounded by beneficial bacteria, it's much more difficult for pathogens to attack the plant roots. So it's a special case. It's a special case because actually you use a plant sucrose, you create a leaven. From that leaven, you create a leaven oligosaccharide, which is probably the main signal in the cascade. So the main signal is a microbial one. It's not a plant one. Yeah. But then it goes back to the plant because at the end, it protects the plant. So if you follow the whole cascade pathway, initially the sucrose exudate at the end is protecting the plant, but through a very indirect mechanism involving beneficial bacteria. So this is the Fructan detour concept. It is essentially a sweet immunity concept because it involves sweet compounds, sucrose, but also leaven oligosaccharides, which are also sweet compounds. And I predict that these leaven oligosaccharides probably also will play a role in intermicrobial communication, because it's not only a matter of bacillus there, it's also a matter of building networks, beneficial networks with beneficial fungi, for instance, trichoderma and so on. And I would not be surprised that this loss would also function either as a prebiotic or as a signal in cross-kingdom kind of interaction, not only with the plant, but also in between different types of bacteria and fungi that altogether might actually build a beneficial network around the plant root. So this is a very interesting and new insight that is coming up, placing potentially fructans in the center of all this business. So this is why, of course, I find it very, very important. So let's jump now, maybe a bit difficult for some, but let's jump to the idea about fructan priming in animals and humans. These workers do not call it priming, they never do. But essentially, you can probably also trigger directly or indirectly some pathways involved in animal and human immune responses. Well, it is for decades that we put adjuvants, inulin as an adjuvant in vaccines. Because we know if you add inulin, the vaccination goes much smoother, much better. So this can even lead to single dose influenza protection, especially in the babies, the neonate puppies and mice and so on. This works very, very strong because they probably rely more than in the adult phase on the innate immunity still, while in the adult phase, the humoral adapted immunity becomes more important. These authors that I mention here, this is a Japanese team, they didn't really publish what fructans, inulins on their own could do, but they have the data and I asked them, and it's amazing because this influenza, of course, piece was there, but also without the influenza, the effects of inulin alone were pretty amazing. So this shows that inulin themselves can actually trigger things, can in a way prime things, put things ready for the defense against certain intruders. So yeah, these direct effects involving toll-like receptors has been described before by a Dutch team. So that is known, the same for leavens and so on. So yes, what is a bit difficult in this story is whether these effects are coming from the short chain fatty acids produced out of the fructans or directly to toll-like receptor effects. In any case, these signaling pathways relying on AMPK, which is actually the alternative of SNRK1 in plants, these are actually put on, and then you have these NFK beta signaling pathways putting on and creating immunomodulatory cascades that actually can help to protect plants, to protect the health. And of course, what happens at the gut system also links to the brain, and these are insights that we got lately. So it's not only affecting our well-being, our body, but also our brain, and this is of course very interesting. Okay, I will end with this research in which they actually combined the intake of higher and lower DP inulins, oral intake with vaccination against hepatitis, and also a control, a placebo. And you see that the antibody titers after 35 days were clearly higher than the placebo for the higher DPs. And also when you look at the TLR2 mediated activation of this NFK beta pathway, you really see effects increasing when you increase the ITF. These are these inulin type fructans, and they are more stronger for the higher TP fructans. So anyway, I conclude with the fact that fructans hold great promise for health promoting applications, not only in plants, but at the same time in animals and humans, that they can directly act on certain receptors probably, or indirectly through microbial effects that then make certain effectors that work well. Fructan priming needs to be further tested in a longer array of plant and animal systems, and also during different developmental stages, and especially further exploration of the type, DP, and dose and frequency of treatment need to be elaborated further. And of course, by what we know now, we need to go deeper in fructan producing biological control organisms, because probably there it's clear that fructans are really acting as signals. And if this is the case, it would be nice, of course, to seed prime with fructans before they enter the soil, so that they get this signal from the very beginning. So, okay, this was more or less my presentation. This is the team that, of course, contributed a lot to all these ideas and data. And I thank you for your attention, and I'm sure you will have questions.